Hi, welcome to today's AP Biology lesson. I'm Allison Kate, and today's lesson is on meiosis and how it generates genetic diversity. So we will be looking at topic 5.1 and 5.2 from the course and exam description, and also 2 science skills, Science Practice 1B, which is explain biological concepts and or processes, and Science Practice 3A, which is to identify or pose a testable question based on observation, data, or a model. And we're also going to be looking at the meiosis part of Lab Investigation 7 from the AP Biology Lab Manual. You should be familiar with concepts of cell division. If you tuned in yesterday, we focused on mitosis and the cell cycle. You should also be familiar with the terms gametes, haploid, diploid, and variation. By the end of today's lesson, you will be able to explain how meiosis results in the transmission of chromosomes from one generation to the next. You should be able to describe similarities and differences between the phases and outcomes of mitosis and meiosis, and explain how the process of meiosis generates genetic diversity. So go ahead and grab a pen and paper. We're going to start with our warm-up, which if you, hang on. Let me tell you that before I get into the warm-up, I do need to make a correction from yesterday's lesson on mitosis. And first of all, I want to give a shout out to a student who emailed me from Florida. Thank you very much for pointing out there is an inconsistency in the math. And then also thank you to all my colleagues in the AP Biology community who pointed out some misunderstandings. And so what I'm going to do is I'm going to do this in 2 phases. The first phase is I want to correct my method for calculating the observed and expected values. Now, I put this together in haste, so hopefully everything is okay here. But what I did is I went to the AP Biology Lab Manual and I screenshotted the how to do the observed and expected values, and then how to go ahead and apply this to your chi-square calculation. And so you can see that this is from the same onion data, and we've got a control and treated data, and then total number of cells. And I actually think the best thing for you guys to do would be to actually go through this and look at it. But I want to just point out how, for one of them, and then you can practice doing the other ones, how you get this expected value for the cells that are in the control or distilled water treatment and the cells that are not dividing. So if you look at how to do that, it tells you here that we're going to take the value in cell A, which if our sample data is here. So that's 148. We're going to add that to the value in cell B, which is here. And then we're going to multiply that by cell A, 148, added to cell C, which is 161, just below it. So you've got A, B, C, and D, and these are your total number of cells. Okay, so if you go through and put all that into your calculator, add it up, divide by the total number of cells, you're going to come out with a value of 126.6, which is rounded up. Use whole numbers when doing your chi-square, and you're going to get 127. And so that is how that expected value is obtained. Go ahead and practice with the control mitosis, the treated interphase, and the treated mitosis. Okay. On Friday, when I'm with you again, I am going to actually have the math set up for you and apply it to the data that I gave you in the lab portion of yesterday's presentation. I was madly working on it this morning, but could not pull it off in time for today's lesson. So stay tuned. All right, so let's go ahead with our warm-up. And if you have been following along with us, our homework is, our warm-up is always the homework from the previous lesson. So yesterday's lesson was on the cell cycle and mitosis, and I simply asked you to identify the 3 major stages of interphase and then describe the sequence in which they occur. So go ahead and jot these down. If you have your homework, go ahead and get it so we can compare to the scoring guidelines. So this seems a little bit repetitive, but if you're going to identify those 3 stages of interphase, you simply need to say G1, S, and G2. Those are the 3 phases of interphase, and that was worth 1 point each. So you can see this was a pretty easy question. The second part is to describe. Oh, and by the way, that identification needed to be in order. So you had to go G1, S, G2. And then the description was also worth 1 point, which is that the cell undergoes generalized growth. Then in the S phase, we synthesize or replicate the DNA. And then the second growth phase is specifically to prepare for mitosis. So that's the description. So that total scoring guideline was worth 4 points. I hope you guys got all 4. So what do you need to know today about meiosis? And keep in mind that one of the outcomes or the things that we want you guys to understand about this is, first of all, what the process of meiosis is, how it contributes to genetic diversity, and then how it is similar yet different from mitosis. So mitosis is a process that ensures the formation of haploid gamete cells in sexually reproducing diploid organisms. It results in daughter cells that have half the number of chromosomes of the parent cells. Meiosis involves 2 rounds of sequential steps that are called meiosis 1 and meiosis 2. But there are some similarities that we want you to notice with mitosis. So like mitosis, meiosis is preceded by a replication of chromosomes. So again, that occurs in the S phase of the cell cycle. So that is representing our duplicated pair of chromosomes. And we've got pairs from our diploid parents, a maternal pair and a paternal pair. And we can say that these are from the paternal side and these are from the maternal side. Meiosis takes place in 2 sets of cell divisions called meiosis 1 and meiosis 2. And we're going to go into the events of these in just a moment. The 2 cell divisions result in 4 daughter cells rather than 2 daughter cells of mitosis. So the end result of mitosis is 2 genetically identical daughter cells. In meiosis, you end up with 4 daughter cells, and they are haploid. They have half the number of chromosomes as the parent cell. So here's a side-by-side comparison. If you look at mitosis, and we're looking at a cell that is diploid, and it has a diploid number of chromosomes as 6. So where we call from yesterday that in prophase, the chromosomes will condense into their familiar chromosome form. These are the duplicated chromosomes. Remember, they duplicated in the S phase. And then those chromosomes line up in metaphase, and then they are separated in anaphase. So you have identical diploid daughter cells at the end of mitosis. If we look at meiosis, there are some other things that we're going to talk about in just a moment, but in prophase, what you're going to see here is homologous chromosomes are pairing up, and they line up in metaphase 1. And in at the end of metaphase 1, we have 2 cells with homologous chromosomes. In meiosis 2, those 2 cells divide again, so we end up with 4 cells when the sister chromatids separate. And the other thing you should notice, if you look at the colors of the haploid chromosomes that are in each of those, or the number of chromosomes that are in each of those haploid cells, they are different. They are different from each other, and they're different from the parent cells that they arose from. So those are some of the key side-by-side comparisons of mitosis and meiosis. Here's a review chart that kind of just puts it all in one place. So if you want to just do a compare contrast, the parent cells are the same chromosome number in both types of cell division. We begin with diploid cells in mitosis and diploid cells in meiosis. The number of DNA replications is the same. DNA only is going to replicate once in interphase in the S phase in both types of cell division. In mitosis, there's a single round of cell division, whereas in meiosis, there are 2 rounds of cell division, meiosis 1 and meiosis 2. In mitosis, there are 2 daughter cells that are produced, whereas in meiosis, there are 4. The chromosome number of the new cells that are produced is the same as the parent in mitosis. Remember, we want genetically identical cells. And then in meiosis, we reduce the chromosome number by half in those 4 cells that are produced. The purpose, as we talked about yesterday, mitosis is for generalized growth, cell repair, and cell reproduction. For meiosis, we want to produce gametes, and in doing so, we want to ensure there is genetic variation in those gametes. So I'm going to take a quick trip through the stages of meiosis. Again, it's really not crucial that you know the exact events that occur in each stage, but there are some key differences in meiosis that I want to point out. So the first thing that I want to point out in meiosis 1 is we're going to separate homologous chromosomes, and that prophase 1 is very different in meiosis than it is in mitosis. So it's typically the longest stage. It takes up about 90% of the time required for meiosis. And just like in mitosis, the chromosomes do begin to condense. However, these homologous chromosomes, one set from mom, one set from dad, right? We've got the maternal and the paternal homologous chromosome pairs. They tend to pair up and twist around one another in a process known as synapsis. And there are 4. So this is called a tetrad, and this will be illustrated in a slide that's coming up. So that's super important, and we're going to talk about the significance of this process in just a moment. Then in metaphase, what happens is those homologous chromosomes line up along the metaphase plate, and just like in mitosis, we have the same apparatus moving those chromosome pairs. We've got our microtubules, which are our kinetochore spindle fibers that are attached to the chromosomes through the kinetochore, and then we have the aster at the end, which is kind of the bracing part of this process, and the non-kinetochore fibers that are not attached to the chromosomes. In anaphase 1, the homologous pairs move to opposite sides of the cell. And then in telophase 1 and cytokinesis, we get those 2 cells, each containing homologous chromosomes. In meiosis 2, it's very similar to what happens in mitosis. Prophase 2 is not like prophase 1. There is no synapsis. There is no exchange of material. And then what happens in metaphase 2, the homologous chromosomes line up in the middle of the cell. And now in anaphase, the chromatids are separated. And so the chromatids move to opposite sides, and you can follow either one of these. It happens in both of the cells that are the result of meiosis 1. And at the end of meiosis 2, you have 4 haploid cells with chromatids or chromosomes that are different from each other and from the parent cells that they came from. Again, it's really important to understand that a similarity is in the mechanism, the spindle mechanism that moves those chromosomes. Okay, so now let's go back and take a look at what happens in prophase 1. So in prophase 1, we've got our non-sister chromatids, the 2 homologous pairs come together in a process known as synapsis, and pieces of that genetic material or gene on a chromosome actually cross over, breaks off, and recombines. The crossover points are known as chiasma. So what you see when you cross over is that the non-sister chromosomes actually exchange segments of DNA. So this is more than one of these crossover points is called a chiasmata, and it just is an X shape is what that chiasma means. So now in anaphase 1, we've got those homologous pairs, and you can see we've got different colors, which represents the exchange of genes or segments of DNA on that chromosome. And then what we'll see in anaphase 2, when those sister chromatids separate, we're going to get different recombinations of those chromosomes. And just a nomenclature, naming thing, the ones that look like the parent, so this one looks like the paternal chromosome, that's called a parental. This one looks like the mother, that's a parental. And then the ones that have different pieces of DNA are the recombinants because they have pieces of DNA from both of those chromosomes. So that is the major event that occurs that makes meiosis 1 different from mitosis. Prophase 1 is where the action is. It's where that extra genetic variation is introduced from the process of crossing over. So now here's the key idea with the genetic diversity. Sexual reproduction in eukaryotes, which involves gamete formation. It includes crossing over, random assortment of chromosomes during meiosis, and then also random fertilization of gametes. All three of these things increase variation, which is the raw material for natural selection. So there are three mechanisms that contribute to genetic variation. The first is that independent of assortments of chromosomes, and I'll show you that in the next image. Crossing over, which we just talked about, occurs in prophase 1 of meiosis 1, and random fertilization, which sperm cell fertilizes which egg is a completely random process. So here's the possibilities of how these chromosomes can line up. If you look at possibility 1, again, I can call out these colors. We've got two homologous pairs. If we say the blue ones are paternal and the red ones are maternal, we've got two different sets of chromosomes, so they can line up in one of two ways. You can see in the first possibility, we have both of the blue ending up in one of the cells at the end of meiosis 1, and both of the red in the other. So that means that our daughter cells are going to have either all blue or all red. If you look at possibility 2, those cells line up. You've got a blue and a red in one cell and a red and a blue in the other cell. And so when those daughter cells are formed, they're going to have a different complement of those chromosomes. So it's important to note that the way that those pairs line up is completely random in metaphase 1 of meiosis. And then in independent assortment, each pair of chromosomes sorts maternal and paternal homologs into daughter cells independently of the other pairs. So it is completely random the way they line up, and they assort independently of other chromosomes. So these numbers are meant to awe and amaze you. So the number of possible combinations when chromosomes assort independently into gametes is 2 to the nth power, where n is a haploid number. So for humans like us, we have 46 chromosomes in a diploid cell, which means we have 23 in a haploid cell or gamete. So that means if you go 2 to the 23rd power, there are over 8 million possible combinations of chromosomes. Then random fertilization adds to genetic variation because any sperm can fuse with any egg. So the fusion of those two gametes, each with about 8.4 million possible chromosome combinations from independent assortment, produces a zygote with any of about 70 trillion diploid combinations. Crossing over adds even more variation. And that is why no other human looks exactly like you. Each zygote has a unique identity. All right, so now hopefully armed with that information about meiosis and how meiosis contributes to genetic diversity, we're going to take a look at the second part of Lab 7, which investigates meiosis. And the objective of this lab is to observe the arrangement of ascospores in the fungus Sordaria fimicola. The spore arrangements in this particular fungus, which is an ascomycete fungus, which means that it produces its spores in sacs or asci, corresponds to the crossing over events that occurred during meiosis when the spores were formed. And hopefully, as we go through this, that will make more sense. So one of our skills is to be able to pose a testable question. So for this particular investigation, a testable question might be, how does the arrangement of spores show if crossing over has occurred during spore formation in Sordaria fimicola? So what happens in this lab is that students will gather evidence to support the claim that the farther the gene for spore color is from the centromere, the greater the number of crossovers will be observed. And now I have a couple of images for you. So what happens in this lab is we propagate this fungus on a Petri dish. This fungus, turns out, comes in two varieties. There's a wild type variety that produces black spores, and then there's a mutant variety that produces tan spores. And we culture them together so that we allow the black spore filaments to reach out to the tan spore filaments, and they actually get together, they exchange their genetic material and reproduce spores. So you get a diploid zygote. I know this is small. I'll show you another image in just a moment. And then they reproduce their spores by meiosis. And then what happens? So these are the four products of meiosis. Okay. Then these spores actually undergo one round of mitosis. So they create genetically identical copies of these spores. So let me just point out that if I've got this black spore, it is going to produce another copy of a black spore. So in this case, I have two products of meiosis, two spores that are black. They're going to each divide. So I'm going to end up with 4 black spores. And the same thing will be true of the tan spores. In this arrangement, I have 2 tan spores. They're going to divide. So I'm going to end up with 4 tan spores. So the overall result of that reproductive event is that I'm going to get 8 total spores. They're haploid. And 4 of them are black and 4 of them are tan. So this is just a drawing showing you how that the ascus, this is the sac, and the ascospores line up. And this whole area here is the fruiting body, or the perithechium. And in the lab, the students are challenged to open up this perithechium in a way that keeps those ascospores in groups of 8. So in other words, you have to use really a gentle technique. You don't want to spread them out like that. So we want to keep them, I always say they kind of look like strands of pearls. We want to keep them in that intact pattern. So here's a little bit more about the process. So you can see that we've got our zygote, and we've got the genes for the spore color on those homologous chromosomes. DNA replicates, and now we've got a duplicated copy of both that tan chromosome and the black chromosome. We're going to look at 2 possibilities, one where no crossing over occurs, and then one where crossing over occurs. And over here, you can see that this is going to be meiosis 1 and meiosis 2, and then the products of mitosis. So if no crossing over occurs, those tan chromosomes and black chromosomes end up in different cells at the end of meiosis 1, and then they will separate into unique gametes at the end of meiosis 2. So you can see we've got gametes that produce tan spores and black spores. When they divide by mitosis, they create a spore pattern of 4 tan and 4 black. Okay. If we look at crossing over, we can see in this image that pieces of the black spore gene are actually going to cross over onto that tan spore chromosome. And so that's the result. Same thing happens with the tan chromosome. We get a little bit of the gene for tan spore color on that black chromosome. And then when they do their first round of division, we can see that they create 2 different cells that when they divide by meiosis 2, we end up with an all tan, a tan with a black tip, which is going to produce a black spore because that's where the gene for spore color is. This is tan. This one has a tan gene and this one has a black gene. So now we have a different pattern and it looks like this. We get 2 after mitosis, 2 tan, 2 black, 2 tan, 2 black. And because of independent assortment, it could also be 2 black, 2 tan, 2 black, 2 tan. So the way to remember this is that a 4-to-4 arrangement is no crossing over, and anything else shows that crossing over occurred. And this is just to show you in real life what those 2 different phenotypes look like. All right, so now let's take a look at the analysis. So we prepare these spore slides by easing those ascospores out of the perithechium, and in no crossing over, we're going to get a 4-to-4 pattern. And if you look, I have an example here. You can see that there are 4 black and 4 tan. So that's no crossover. Any other possibility is crossing over. And so I gave you the 2, 2, 2, 2, but you could also have 2 tan, 4 black, 2 tan, or vice versa. So anything other than 4, 4 is going to give you a crossover pattern. And you can see here's an example of this. So 2, 4, 2. And so students go about counting and recording their results. This is a data table from the lab manual, and you record the number of ASCII. It's a really good idea to count up to 50. More is better because you're going to get closer to the expected value. And so in this case, we can see that students have counted 24 non-crossovers and then 26 crossovers. So then what you can see is we've got, we want to calculate the recombination frequency. So that's how many actually crossed over out of the total number of ASCII. And so that's 26 out of 50, or 52%. Now remember that to figure out where that gene to centromere distance is, and remember that we are trying to find data to support the claim that the farther away that gene is from the centromere, the more crossovers will occur. We need to divide this by 2. And the reason we need to divide it by 2 is that remember that there is a mitotic division that occurs. So map units, which are kind of just arbitrary units to show you where that gene for spore color is on that chromosome, we need to divide that by 2. So we can see that the calculated gene to centromere distance is about 26 map units. Okay, so you can also remember that the recombination frequency tells us how far, this should be far, the gene for spore color is from the centromere. Data that is close to 26 map units will support the claim that the farther the gene for spore color is from the centromere, the greater the crossover frequency. You can also do a chi-square or goodness of fit test for this data. This one is a little bit different because we know that our expected value, if we count 50, should be 50% crossover, no crossover. So you can just go ahead and do your O minus E calculation for chi-square that we went over yesterday. From the critical value table, we know that we use a probability of 0.05. We still have a degree of freedom of 1 because there are 2 categories, crossover, no crossover. Degree of freedom is the number of categories minus 1. And so our critical value from that critical value table is 3.85. Now, we remember that if our chi-square value is less than our critical value, we're going to accept the null hypothesis. And in this case, we're going to fail to reject the null, which means accept, and that means that there's no difference between the observed and our expected results. And we were spot on with the map units of 26 map units. Okay. So now let's look at some FRQs. So this first FRQ is a meiosis FRQ, and I'm going to go over how we get this answer in the scoring guidelines, because this is one of those common misunderstandings that students have. So we've got a cell, we've got chromosome 1 and chromosome 2, and it tells us that in a certain species of plant, the diploid number of chromosomes is 4. So we've got 2n equals 4. Flower color is controlled by a single gene in which the green allele, which is represented, it's dominant, right? So it's got the capital G. It's dominant to the purple allele, which is going to be lowercase g. Plant height is controlled by a different gene in which the dwarf allele, capital D, is dominant to the tall allele, little d. Individuals of the parental generation, which is abbreviated P generation, with the genotypes big G, big G, big D, and little g, little g, little d, were crossed to produce F1 progeny. Construct a diagram below in the template to depict the 4 possible normal products of meiosis that would be produced by the F1 progeny. Show the chromosomes and the alleles they carry. Assume the genes are located on different chromosomes and that the gene for flower color is on chromosome 1. So going back up to chromosome 1, this is going to be the color. So it's either going to be big G or little g, and this is for height. So it's either going to be big D or little d. Now remember, you have to figure out the first cross. So you got your F1 progeny, then you have to figure out their gametes. And this is always a challenge for students. So I want to give you a minute to think about it. And then let's go ahead and look at the scoring guidelines. So if you look at the scoring guidelines, I've labeled these gametes. We've got gamete 1, gamete 2, gamete 3, and gamete 4. And you need to have those particular gamete combinations. And I'm going to show you how you get it. So the diagram must include unduplicated chromosomes, so a single chromosome with either a capital G or a lowercase g, and one unduplicated chromosome for the height allele. So how did we figure this out, right? So if the parental generation is crossed to get the F1 generation, they are heterozygous at each trait. And that's because, let's call this mom, she only has the dominant allele for both color. So this is going to be green and dwarf. And then dad is going to be purple and tall. And because they only have those alleles, they're going to be heterozygous at each trait for each trait. And so this would be a green dwarf F1. But now we need to figure out what the F1 gametes are going to be. And you're going to borrow something that you probably first learned in math class. You're going to use FOIL. So the first two are going to be big G, big D, and that's gamete 1. Your second two or outer two is going to be big G, little d, and that's gamete 3. Inner two is going to be big G, little g, big D, and the last two is little g, little d, and that's gamete 4. So make sure you follow that. That is the only way you can determine how those gametes are going to end up. Okay, another practice with diversity. So a research team has genetically engineered a strain of fruit flies to eliminate errors during DNA replication. The team claims that this will eliminate genetic variation in the engineered flies. A second research team claims that eliminating errors during DNA replication will not entirely eliminate genetic variation in the engineered flies. Describe one mechanism that could lead to genetic variation in the engineered strain of flies. Okay, so I gave you the top 3, but there's actually more. So you only need to pick one mechanism. So think about that, and let's look at the scoring guidelines. Any one of these bullet points would earn you that point. So sexual reproduction produces offspring with new combinations of alleles or traits. Fruit flies have fewer chromosomes than humans, so it's not 70 trillion possible combinations, but it's still many, many new combinations. Crossing over produces even more new combinations of alleles and traits. Independent assortment produces new combinations of alleles or traits. Random fertilization produces new combinations of alleles or traits. So those were the big take-home lessons from today's presentation. But you could also imagine that immigration or gene flow could introduce new alleles into this genetically engineered fly population. There could be a viral infection that inserts DNA into the fly genome. Non-disjunction, which is the failure of chromosomes to separate properly, could cause an anomaly in chromosome number. In other words, you end up with too many or too few. Chromosomal rearrangements could inactivate genes or result in multiple copies of genes, so things like duplications or deletions. And then there could also be some type of a mutation that could be introduced into the DNA. So any one of those mechanisms would introduce genetic variation. And remember, this is a key idea for meiosis, that it introduces genetic variation, which is the raw material for natural selection to act on. All right, so let's do a check for understanding with skill 1B, which is to explain biological concepts and or processes. So this is a former FRQ from a former exam, and it is going to actually give you a compare contrast type thing between the two types of cell division that we've discussed yesterday and today. Both mitosis and meiosis are forms of cell division that produce daughter cells containing genetic information from the parent cell. You are first asked to describe 2 events that are common to both mitosis and meiosis that ensure the resulting daughter cells inherit the appropriate number of chromosomes. Think about how they get moved around. And then in B, the genetic composition of daughter cells produced by mitosis differs from that of the daughter cells that are produced by meiosis, and then describe 2 features of cell division that leads to these differences. I want to remind you of something before you try to answer this. Remember this? So I showed you this before. This is kind of the nuts and bolts or the compare contrast slide. So I want you to think about that as you begin to answer this question. Okay. So scoring guidelines for part A, you need 2 events. So each bullet point is 1 point and it's 2 points maximum. So remember, how do the chromosomes move? It's that spindle apparatus. The microtubules will attach to the chromosomes and move them in both meiosis and mitosis. In both cases, in prophase 1 of meiosis and in prophase of mitosis, the chromatin condenses into chromosomes. We have in both types of division the alignment of chromosomes across the center of the cell in metaphase before they separate in anaphase. And the separation of those chromosomes and chromatids into the daughter cells occurs both in mitosis and in meiosis 2. We regulate the cell cycle the same in both types of cell division. So that checkpoint regulation occurs, I explained it in mitosis yesterday, but it occurs in meiosis as well. The replication or synthesis of DNA occurs in the S phase in both types of division, and cytokinesis occurs to separate those daughter cells after either mitosis or meiosis. And then in part B, there's that table, right? So 2 features of the cell division process that lead to these differences. For mitosis, there's a single round of division with 2 cells resulting. For meiosis, there's 2 rounds of division with 4 cells resulting. For mitosis, the chromosome number is the same as the parent cell. Remember, genetically identical cells, so it's diploid to diploid. And then in meiosis, we have that reduction division. So we reduce the chromosome number by half. So in your gametes, you have 23 chromosomes compared to the precursor cells that they arise from, which is 46. Chromatids separate in mitosis, and they only separate in mitosis 2 because it's the homologous chromosomes that are separating in meiosis 1. Crossing over only occurs in meiosis, and homologous chromosomes assort independently in meiosis. Okay, so hopefully you got all 4 points on that one. Okay, so here's a sample answer that you can take a look at. That was a stellar answer. The separation of sister chromatids in both mitosis and meiosis 2 ensure that each daughter cell receives the appropriate number of chromosomes. Also, the lining up of chromosomes along the middle of the cell ensures the chromosomes will separate properly. And then in 4B, unlike mitosis, meiosis results in 4 haploid cells rather than 2 diploid cells because cell division occurs twice to produce gametes. Also, the chromosomes in the cells that have undergone meiosis are recombinants of each other because of the synapsing that occurs in prophase 1. And this happens unlike synapsing, or synapsis does not occur in mitosis. So you can see we don't take points off for a new use of a new form of the word synapse. Okay, and so that was a perfect 4 score. All right, so your task verb this time was describe, and so you want to paint a picture using words. You want to describe it as if someone cannot see it. So here's your debrief. Your learning objectives. You should now be able to explain how meiosis results in the transmission of chromosomes from one generation to the next. You should be able to describe similarities and differences between the phases and outcomes of mitosis and meiosis. You should be able to explain how the process of meiosis generates genetic diversity. Remember those big 3. You should be able to explain biological concepts and or processes, and you should be able to identify or pose a testable question based on an observation, data, or a model. And again, just like yesterday's key takeaway, heritable information provides the continuity of life. So just some common errors and misunderstandings. I told you about how students have a difficult time finding the gametes of the F1 progeny. Remember FOIL, okay? So the good news is that students were able to articulate correct information about both meiosis and mitosis. The bad news is that students equated mitosis and meiosis with the entire cell cycle, not just nuclear division. So keep that in mind, that that is just a very small portion of the cell cycle. Many students were unclear about the events that occur specifically during the nuclear division. Many identified chromosome replication or DNA synthesis is happening during mitosis or meiosis, and I hope that you've noticed that I have stressed both yesterday and today that that synthesis or duplication, replication of chromosomes happens prior to nuclear division. Many students fail to identify spindle fiber formation as a distinct event in mitosis or meiosis. And remember that spindle fiber apparatus is how the chromosomes are moved during the process of nuclear division. And while many students were aware that checkpoints exist, they were not clear as to their purpose or why they occur. Many students erroneously identified homologous chromosomes as lining up during mitosis or mitosis 2 rather than meiosis 1, as is the case. And students often confused haploid cells and diploid cells. So try to keep all of those things straight when thinking about these cell division processes. And I know that they're similar. I try to teach them completely separate. So first you learn mitosis, go on to some other things, and then come back to meiosis so that you're not confusing them. So that may be a strategy to keep them straight. All right, so here's your homework, and this will be debriefed tomorrow as your warm-up, and tomorrow you'll be with Miss Pfizer. She's going to be talking about Mendelian genetics. So that will kind of solidify some of these events of meiosis for you. Meiosis occurs in many organisms as part of sexual reproduction. Describe 3 differences between mitosis and meiosis. And remember, if you need to, you can go back and look at that review slide. So I also want to end by reminding you that you have a to-do list, your exam prep checklist. The exam is going to be on Monday, May 18th at 2 p.m. Eastern Standard Time. At that time, you're going to need to have a device to take the exam on. You're also going to need to have internet connectivity. If you need help with either of these things, please reach out to College Board at cb.org/tech. And you need to do this by this Friday, April 24th. You also need to be certain to check that the College Board has your correct email address. They're going to be emailing you your exam ticket. You will also be able to access your exam ticket from your student side of AP Central. So there'll be a couple of ways to get it. If for some reason they miss your email address, they send it to the wrong email, or you have a proxy wall that prevents you from receiving that email. And then finally, please let your AP Bio teacher in on your plan, whether you're going to test or not test. No judgment, just want to know who's going to be testing. And finally, we will be doing, so our last live session will be on Friday, May 1st. The exam isn't until May 18th, but AP exams do start the week of May 11th. So in that gap week, we are going to be doing some exam prep things. We're going to have some practice questions for you. We'll debrief those practice questions with you. And also we'll make sure that you have some practice understanding how to upload your responses. So we hope you'll join us for that week as well. So thank you so much for watching. Make sure you do your practice question. That will be your warm-up tomorrow. And as I said, your next lesson will be with Miss Pfizer, looking at Mendelian genetics. Have a great rest of your day. And one more thing, I will have a corrected chi-square analysis for you from the lab investigation 7 when I see you again on Friday. All right, have a great day. Thanks for joining.